Real pleasure to introduce Dr. Ari Hakimi as today's Grand Round speaker. He's an associate professor and co-leader of the Translational Kidney Cancer Program at Memorial Sloan Kettering Cancer Center. Dr. Hakimi is a urologic surgeon whose focus is on the care of patients with urologic malignancies, especially kidney tumors. He received his medical degree and residency training from Einstein College of Medicine and completed his fellowship in urologic oncology at Memorial Sloan Kettering Cancer Center. His research really aims to understand immune infiltration, inflammation, and the tumor microenvironment in RCC, and to identify novel therapeutic targets to overcome resistance to systemic therapy. His studies apply bulk, single-cell, and spatial RNA sequencing, flow cytometry, and immunogenomic analyses, really to understand both patient samples and a novel immunocompetent kidney cancer mouse line that his lab has developed. Really has been a game changer for the study of kidney cancer and particularly the immunobiology of kidney cancer. I followed Ari's work for many, many years now, and to see him go from what was a rising star in kidney cancer research to now really one of the world leaders has been a pleasure, and it's a body of work that I've tremendously admired. So it's really a pleasure to be able to welcome Dr. Hakimi to Grand Rounds today.
All right, thank you so much. And it's really a pleasure to be here at Yale. And especially, I was especially enthusiastic to come here because of David. And I mean, talk about rising stars. David is incredible, and he's got great mentors here with Harriet and others. And I think it's a real pleasure here. So that's my only disclosures, none of it which is pertinent to this talk today. So I'll talk a little bit about the genomic and genetic background of kidney cancer. In particular, clear cell renal cell carcinoma, which is the most common and aggressive form of kidney cancer, but also one of the most immunoresponsive. We'll talk a little bit about the role of the microenvironment as a predictor of response, and really focus on myeloid compartment, which is one of my lab's interests. A little bit from the genomic determinants of this, and then some of the insights using both human and mouse strategies to understand this for future targeting. So kidney cancer is the sixth most common cancer overall, or eighth most common cancer overall, sixth most common in men. There's a 2-to-1 gender difference. And we know that, within the kidney, there are many, many subtypes of kidney cancer. And even if you just took the most common subtypes, they're very genetically different. Clear cell represents the most common form. About 65–70% of all kidney tumors are clear cell. And we know the most about it from a genetic standpoint. But there's also some very intriguing phenomena that exist in it, particularly the amount of immune response. And it's still not clear why these tumors are so immune infiltrated and why they are so responsive to immunotherapy compared to other tumors that are much more highly mutated, for example, like melanomas or bladder or lung cancers, where you see a lot of mutations in those tumors. And probably, everyone references TCGA papers initially in terms of the fundamental understanding. And I think some of the takeaways from this analysis, which was one of the first tumors to be profiled, was that it's really dominated by a few driver mutations related to tumor suppressors on the 3P locus. There's not a lot of mutations. There's some copy number events that are really fundamental, and maybe some of which are enriched in metastases. But there's not an obvious clue as to why these tumors retain such a high immune infiltration. We also know a little bit about what happens thanks to seminal work from the Sanger Institute, where they looked at what were the fundamental events that are associated with clear cell renal cell carcinoma development. And what this paper showed for the first time was that the loss of chromosome 3P, one arm, is critical to the oncogenesis, and then that's followed by a VHL loss, whether it's mutations or methylation, but basically 90% of all clear cells have this. And then eventually over time, additional driver mutations are lost, and that leads to different evolutionary subtypes. And in this paper, Samra Tarajlik and others came up with a relatively complex schema, but fundamentally you can think about it as the tumors lose VHL, and then they usually acquire one or two additional hits to form into sort of different trajectories. We know that tumors that lose PBRM1 and SETD2, for example, may be more angiogenic. They may tend to be a little bit more indolent overall, maybe possibly more responsive to certain therapies, including even immunotherapy, although that's not entirely clear. And then BAP1 mutations, which occur as well, are typically associated with more high-grade, aggressive, proliferative tumor types. And then you can have multiple clonal drivers, which also represent a very aggressive form of kidney cancer. So we're starting to get a better framework for the underlying genomics, but none of this really has been shown to be targetable. So for years, we just really started to understand what was driving kidney cancer, but we really wasn't giving us any further insights. It weren't an oncogene that you could develop a target to. And while people are certainly working on epigenetic regulation and strategies, it's certainly not an obvious pathway forward in kidney cancer, at least right now. And at the same time, we also knew clinically that most of the targeted therapies were limited to the microenvironment of the cancer. And really, we've seen a tremendous growth in outcomes and survival for kidney cancer patients, but it's all really focusing on the microenvironment. So even back in the 90s, when we were studying IL-2, both in melanomas and kidney cancers, that was really the only treatment that seemed to work. We had tried all the chemotherapies you can imagine in the 80s, 90s, and you would see responses, even 7 to 10% cures, but with very, very high toxicity in those populations and in those patients. And it wasn't until the advent of really by work from Bill Kalin and others where we recognized the importance of HIF that all these focus on VEGF had come around. And then it wasn't really until this notion of immunotherapy came around that we started to see this big revolution in terms of survival and outcomes. But it's all really focused on the microenvironment. We know that you can take some of these genetic events that I mentioned earlier and risk stratify patients a little further. This is work that we did several years ago now, looking at the impact of some of these common mutations in outcomes for patients that were receiving VEGF therapy. And it was, prognostic. Maybe you could further stratify patients that were grouped into clinical risk groups by common mutations, but it wasn't really telling us anything about the underlying immunobiology or angiogenic biology in these tumors. It was really more just a prognostic feature about it. So with this revolution of immunotherapies, both by themselves and in combination with VEGF therapy, we've seen the survival rate and the response rates go up dramatically. So, the median survival when I first started my training for metastatic kidney cancer was, a year and a half or so, and now we're pushing 5 years for a lot of patients, and potentially curing some patients. And there's a real need to understand why that's the case and how we can do better, because we know that invariably most patients, despite this high response rate, will eventually develop resistance. Understanding why that's the case requires, good models to do so. We know that from an immunotherapy standpoint that it's not a TMB-driven tumor, at least not obviously, and maybe you can break down the types of mutations a little bit more. I was just talking to David about this last night, but it's not obvious. There have been several attempts to look at TMB as a predictor for responses, and most of the large clinical trials that have been performed that have released their data have not shown this, and certainly David has been on the forefront of this. But if you look across some of the major phase 3 trials that have at least released their data, there's not really a signal at all with respect to tumor mutation burden. We've looked David and others have looked at whether mutations in PBRM1 or loss of 9p, which is a common event in metastatic kidney cancers, whether that's associated with immune infiltration patterns. There may be some signals there. It's not a clear biomarker, though, and that sort of has been lacking from a mutational and copy number standpoint. So I think one of the things that is unique about kidney cancer is that we've started to look many years ago now at transcriptomic predictors, because the mutations are clearly, and copy number events are clearly not sufficient to determine who's going to respond, at least from a biomarker standpoint. And you can just take a very simple metric of the uniqueness of kidney cancer. And this, I just plot out, VEGF-A and CD8 infiltration. This was an older slide, but I like showing it because I think it shows the uniqueness of kidney cancer, clear cell at least, with respect to some of the microenvironmental genes. And we know that they're just dominated by high infiltration of CD8 cells and high angiogenic programs. So the question, of course, and this was shown across cancers, and it's really distinct from the normal tissue. If you look at, for example, lung cancers, much of the lung itself is very immune infiltrated, likely due to smoking or other carcinogenic features. But the kidney itself, the normal kidney, is not particularly immune infiltrated, but the tumors are often very dramatically infiltrated. So there's something very distinct about the actual tumor itself rather than the underlying organ that it's derived from. This was work we did when I was just starting out, and we used immune deconvolution strategies to show this. You could take signatures for T cells or for macrophages, NK cells, et cetera, and you could start just deconvoluting bulk RNA sequencing data and start to try to understand where tumors or particular samples might fall in a spectrum. And you can use this to also subgroup. So we use this strategy to kind of think about tumor microenvironmental subgroups within kidney cancer. And this was our first attempt about 8 or 9 years ago to look at, whether there's these enriched groups and whether there's, other groups within kidney cancer. Maybe that would sort of explain why you see some really great responses in this tumor type. And we did see that. You could clearly see these T cell infiltrated clusters. You could see at this point, we really didn't think about angiogenesis, but in retrospect, you've seen angiogenic cluster, and we'll talk more about that in a minute. And it did correlate with certain genetic programs, mostly antigen presenting machinery programs. So there was an upregulation of antigen presenting machinery transcript, but it wasn't clear still from this point what was actually driving this. We looked at genetics, common mutations, and wasn't at least obvious at the time when we first did this study, although that's evolved a bit. And that same approach was applied by Genentech when they first published and analyzed the emotion trial. This was the first attempt to combine VEGF and IO therapies. They used tislelizumab and bevacizumab, which is a VEGF monoclonal antibody. And this trial was negative in terms of improving the standard of care, but it was biomarker rich. And I give a lot of credit to Genentech for not only doing phenomenal genomic work, but also making it all publicly available, which is something that other companies have been a little reluctant to do so. And maybe it was because it was a negative study they were willing to share so much, but it really was very helpful. And David McDermott and others from Boston performed really the first type of analysis in the context of systemic therapy to show that these microenvironmental groups, angiogenesis and T cell infiltration and myeloid programs, may stratify patients into different groups, but also may associate with response. And one thing I would point out, I think it's sort of logical that a tumor that may have a lot of T effector cells would respond well to immunotherapy. But what they also showed was that myeloid populations, as determined by, again, a gene program, were driving resistance also. So you could be a T effector high tumor, but if you had a high myeloid program, it could supersede that impact and in fact actually show dramatically different responses in that context. So that really sort of laid the groundwork for not only the microenvironment relevant, but also it could predict responses and maybe give us some insight into resistance. We applied that same strategy initially to just a VEGF cohort only, and this was work that I did with Bob Mocher, one of my mentors at Sloan Kettering for many years. And this was just looking at the first trial that compared 2 different VEGF inhibitors. At the time, we had really sunitinib, and this was the first attempt to try a different strategy or a different VEGF inhibitor. And, it was really more to look at tolerability. There was no difference really in responses, but actually pazopanib at this, showed a better toxicity profile for patients. So that became the standard of care, at least in Memorial, for many years, until obviously we developed next generations in immunotherapies. But at this time, we took a transcriptomic approach. So we had microarray data from Novartis, and we had looked at this question of whether you can identify subgroups, and we found, 4 transcriptomic subgroups at the time. They tended to really stratify patients. Again, all these patients receive VEGF first line, so clean cohort. And you could clearly see a difference in groups. And there was a green group here that was very responsive and a red group that was very not responsive. And then there was these yellow and blue in the middle. And the green group really had a lot of angiogenic program, and that made sense, right? If you have a lot of angiogenesis, it makes sense that you would respond very well. But the red group, which was number 4, that was the one that didn't respond well. They were actually the worst, but they had the second highest amount of angiogenesis. So why was that the case? Why didn't they stratify nicely by angiogenic program? And when we compared that group to the other groups, we could see that it was really being dominated by a myeloid program. So despite having high angiogenic phenotype or transcript, they were reverse in their response based on an infiltration of myeloids, at least inferred by microarray data. So that suggested that it could actually be driving a response overall, and the microenvironment may be useful in understanding biomarkers in metastatic kidney cancer. And then actually, this was something that we did at the end. And actually, my fellow at the time, one of the urology fellows at the time who was working in my group, actually had the suggestion that we kind of lumped them all together because pazopanib and sunitinib were sort of both VEGF inhibitors, but we know that the TKIs target lots of different kinases. They're not super specific. And actually, if you look at the macrophage and angiogenic groups, you could actually see that pazopanib has quite a different stratification than sunitinib. This suggested to us, we didn't talk about it too much in the paper, but it really suggested that the targets of these TKIs may also actually be driving some of their responses. So some of these kinases are present on immune cell populations, for example. And the fact that these biomarkers were actually different with respect to the different TKIs was something that we've now followed up on. And I think it's a really interesting finding that he made. And this just leads back to the same concept that what Dave showed in this beautiful paper from the Genentech study. Others have looked at microenvironmental features in other more positive trials. So this was also sort of a negative, well, not negative, but has not been brought forward further. This was the Javelin 101 study, again, avelumab and axitinib. Again, another combination of PD-1 and VEGF. And they focused on a lymphocytic signature, identified 26 genes. Again, specific signature for a specific trial. Some of the challenges of these have been, applying it to other data sets. But again, you could see the microenvironmental features being associated with response here, and suggesting, that we could utilize this as a strategy. And then there have been subsequent efforts by collaborative groups, including Genentech, again, to integrate what we know about mutations and those evolutionary subtypes I showed you earlier and the microenvironmental features. So if we have the microenvironment and we know the genetics that arise as kidney cancers evolve, could they be sort of grouped together to form these kind of different molecular subgroups? And I think there's been some attempt to do this. I think it's improving, but you sort of have a sense that there are these different angiogenic stromal and angiogenic alone. So some of these may have this myeloid phenotype that I showed you earlier and just a purely angiogenic tumor. Maybe the purely angiogenic tumors would respond really well to VEGF alone. And those tend to be the less aggressive tumors, PBR1 mutated. And then you have the ones that are myeloid and angiogenic, and those actually don't respond at all to VEGF inhibitors. And then you have these proliferative ones and other ones. So we're starting to get a sense that maybe you could subgroup kidney cancers into those features. And then came along this notion of single cell. And there have been a series of papers, one of which David led, but that came out from the time because we had done all of this work on bulk RNA sequencing. And as the fields across cancers have evolved, we started utilizing single cell to not only get a better sense of what was happening, but also really understand what are the specific features of these of the microenvironment in a much more high resolution. So the advantage of bulk RNA-seq, of course, is that you can do big numbers of samples because it's relatively inexpensive. Single cell gives you deep dive, but often the cohorts were much more modest in size. So I think there's constantly a need to go back and forth. If you find a signal in one, you have to validate it in the other, so to speak. And so we did this in clear cell, focusing really on patients that had received just dual immunotherapy with PD-1 and CTLA-4. We focused on 6 patients initially when we did this together with Ming Li, one of my immunology mentors, Christina Leslie, who's a computational biologist and a very, very talented graduate student at the time who's finishing his postdoc at Harvard now, Chirag Krishna. And we looked at patients that had either were high risk and had not received PD-1 right away, or versus ones that had Ipi-Nivo and eventually went on to surgery. One of the unique things I do as a surgeon is that we're able to get tissue after treatment, and kidney cancer has evolved so much so because of the response rates now to upfront immunotherapy that most patients, if they come in with metastatic disease, will get upfront systemic therapy, and then we're being asked to operate on them later on. So that gives us a unique opportunity to study tissue after treatment, which is something I think really unique to kidney cancer. Amongst many solid tumors, we have this opportunity. So we were able to utilize that strategy here. And this sort of gave us a broader sense, and this has been replicated, I think, by many other single cell studies, including David's really seminal work in this. And you can kind of get a sense of what's happening. Now, one of the things that's interesting about a quark of single cell is that, there's, for those of you familiar with the technology, is that, there's generally at least if you do single cell and not single nucleus, you have to do some sort of sorting. And a lot of the tumor cell populations actually die off from that process. They're very fragile, for some, ironically, and immune cells will often survive, although you lose neutrophils. So kind of interesting quirk of any sort of single cell study that you do, you lose a lot of the cancer cell populations, but we're able to kind of get a good sense of what's going on in the immune cell population. And you get a general sense that, and this has been replicated by our flow analyses over the many years, that about 40 to 60% of the immune compartment is made up of T cells, and you have a good amount of TAMs in this, and then a whole bunch of other populations, including B cells, NK cells, and dendritic cell populations. But they're really dominated by these T cell and TAM populations. And you could further phenotype them into, and this has been done, everyone's got their own slightly different way of phenotyping populations, but this allows us to sort of get a sense of what's happening in both primary sensitivity and primary resistant patients. And, this one, again, we had ipi-nivo resistant and a mixed response and a complete response. Complete response patients are always interesting because why are we operating on them if they have a complete response? Well, when I say complete response, I mean that the tumor's mass itself is not viable. There's a mass there, we take it out, but actually under the microscope, there's no tumor left. It's just a conglomerate of immune cells and fibroblasts. And so that's kind of an interesting population to look at because what's residual there. And there we found these tissue resident T cell populations that were very abundant in the residual mass of that kidney, even though there was no tumor left. And then we found within the patients that were non-responding, it really, there was T cells there, but it was really dominated by specific TAM populations. This was a primary resistant patient. He had big tumor, big lymph nodes. We gave him a trial with ipi-nivo to see if that would help, and it didn't. So we still ended up operating on him. No response in the tumor whatsoever. And you could see this was really a TAM-dominated tumor type. So it started giving us insights that really this may be associated, again, small numbers. So you really have to start building that out. And you can develop signatures, which is what we did. We actually took the single cell genes and from the different clusters and overlaid them on some of these genomic signatures that have been published. The Javelin one I mentioned, the genomic ones from Genentech, and we started saying, like, what are the actual populations that they're capturing? You get a better sense that there are some dominant TAM populations, and that some of these populations may be potentially targetable. And I'll talk about that in a moment, but I want to bring your attention to some of these adenosine signatures that were published from an A2AR inhibitor, which is something that has been shown to potentially shift TAM phenotypes. So this was sort of an interesting way for us to look at the data, and you could develop signatures based on the single cell data and compare them to existing signatures to see if you could further stratify patients and responses across different data sets. And we were able to do that. And then the question is also, well, is there a relationship between the underlying genetic microenvironment and these specific immune microenvironments? So I showed you again on bulk, maybe there's these different subclassifications, but we also know there's a lot of heterogeneity in kidney tumors, right? So we know that if you took a kidney tumor and you sequence different regions, Charlie Swanton showed this many years ago in a famous paper in New England Journal, intratumoral heterogeneity exists. Does that same thing apply to the microenvironment as well? And that's something, of course, if you're going to develop a biomarker or suggest something, you have to think about that. And this is work that we did in collaboration with Illumina, where we really thought about this question of, okay, now we have a good sense of what's going on in the microenvironment. We have a good sense of what's going on in the underlying genomics, and how does that relate to the individual tumor? And one of the reasons why clinically that's interesting is because if you look at data sets where the primary tumor is still in place with the patient with metastatic disease, and they get immunotherapy, often the Mets will respond well, but the primary tumors maybe only shrink modestly, right? So there's not that same level of response. And one hypothesis is that, well, it's because the primary tumor is more clonally diverse, and the Mets is just a clone that was able to metastasize out, that was selected for. So when you get a response in the Mets, maybe it's because there's just a clone that's really responsive, but the primary may only have that clone in part of it. And it's sort of been our rationalization for continuing to operate on these patients, because I tell them, well, I like operating, but more fundamentally, it's actually because we think that we're removing the biological diversity of them, even if they've had a good response up front, the chance for them to develop resistance down the road may come from the primary. And so we tried to look at this with multi-regional sequencing, again, relatively modest numbers, but we utilized the combinations of DNA and RNA and TCR and different things within tumors that had been exposed to immunotherapies as part of a trial that we ran and others. And so we were able to look at the question of whether overall immune diversity is associated with, overall genetic diversity is associated with particular microenvironmental phenotypes. Indeed, we found, at least in this study, that if you were a very highly diverse tumor from a genomic standpoint, you were more likely to be a myeloid high tumor, which was interesting, and vice versa with respect to some of the antigen presenting machinery genes. So the ITH tumors were actually lower with respect to the APM genes. And actually, if you took a specific tumor and you actually marked out the immune infiltration patterns, you could start seeing evolution within that same tumor. So this is an example of a tumor that was ITH high. It had a lot of intratumoral heterogeneity. It was the PBAR1-SETD2 kind of microenvironment or evolutionary subtype. And we were able to look at individually in different regions of this tumor to see, we found that some of the regions were very T-cell infiltrated, at least by RNA, and some of them were very myeloid infiltrated. And we were actually able to track down like what were the individual genetic events that were occurring as this tumor developed. And you could see that, as the tumor developed, there was loss of HLA and maybe some CDKN2A and B loss, which has been associated with a more immune desert or less immune infiltrated microenvironment. So within the same tumor itself, you could see this evolution, and that was correlating with the microenvironmental features, suggesting that there's this constant interplay. And I think David has shown this, and others have suggested this constant interplay between the underlying genomic architecture of a tumor and what's actually, underlying the response microenvironmentally in that tumor. Obviously, we don't fully tease out the mechanism here at all, but it begs the question that there's an opportunity here to explore this. And what about in the metastatic question? So that was another question we had in the lab. So I've showed you everything in terms of treatment response potentially, but we also wanted to know, are these microenvironmental groups also predicting or associating with development of metastasis, which is a different question, right? You could have a microenvironment that's really important for treatment response, but it may not be associated with metastatic development. So David had somewhat hinted at this with his work with Ellie and Tony and others at Dana-Farber, and they showed it, very elegantly in this paper that looked. Our paper focused on the advanced disease in ipi/nivo treated, but David's work was performing single cell sequencing on early locally advanced and metastatic tumors, and at least in this work, he showed this evidence of T cell exhaustion, but also this shift in the macrophage polarity as tumors became more aggressive, more metastatic. So suggesting to us and others that, maybe some of these TAMs and myeloid populations that were so driving responses are also associated with metastatic development. And so for this, we again utilize our strategy with going to clinical trials, and we worked with this adjuvant study. So this was one of the series of negative studies, unfortunately, but again, the benefit of having a lot of genomic data, looking at whether giving a VEGF inhibitor monotherapy, again, pazopanib in this case, was associated with better outcomes in patients with high-risk non-metastatic kidney cancer. All these patients in this trial had advanced kidney cancers. They had a high risk of relapse, but standard at the time was just to observe them. And so there was a series of trials to see if you gave a VEGF inhibitor, whether that actually was improve their survival. The vast majority of the studies were negative. One was sort of positive, but no one has really adopted it. And now we've moved on to immunotherapy. But at this time, this was a very interesting study. So we had microarray data again from Novartis. We compared again, all high risk. So you're sort of controlling for the potential tumor confounding features, right? They're all high risk patients. And we compared the ones that relapsed versus the ones that didn't. This was work that one of our fellows led, and who's now at Rochester with a surgeon scientist track there. He's a great guy, Phil Rapolt, with Lin Vuong from my lab, who's a senior member now. And we compared the tumors that recurred versus didn't, and we used an unbiased, whole genome approach with this. And so, of course, you saw things like EMT and mTOR, which made sense to us because those are obviously very known and relevant oncogenic processes that promote metastases. But we also saw a lot of immune inflammatory genes, in particular IL-6 and JAK and STAT3 kind of stood out to us as being a driver of metastasis. Again, we applied the same single cell strategies where we had done before to see, what are these myeloid inflammation and IL-6 pathways, what are they really converging on? And indeed, it showed a real enrichment in some of these TAM populations that we had defined a little bit better with the single cell data, and suggesting that if you were a tumor that was myeloid high or adenosine high, similar overlapping signatures, you are more likely to develop metastasis. Again, controlling for other features, all of the clinical and pathologic features. These are completely independent programs. And you could show that if you were a MSK inflammatory signature, which was a gene signature we developed from the microarray data, strongly overlapping with the myeloid signature from Genentech and others, that you could take all these high-risk patients and really, it's pretty amazing to see curves like this separate out. But again, all of these patients, this is a myeloid low tumor, intermediate, and then very high, and you could see their survival probability. And then we were able to replicate this in multiple other data sets, including from one of our former fellows who's at Moffitt now. And again, showing that if you were controlling for all these high-risk features, from a clinical pathologic standpoint, you could still stratify patients by the risk of recurrence in that. And it didn't seem to be associated much with the T-cell response. So what was driving metastasis, at least in this data set, but again, it has been validated in others, was not really a T-cell driven process. What was, at least from a microenvironmental standpoint, promoting metastasis was independent. We tried all the different T-cell signatures that had been shown. We looked at IHC scores. We had IHC from CD8 infiltration patterns here. We were able to see if they were inflamed or excluded. And we really didn't see, maybe there's a signal with the desert. Those are the tumors that have no T cells at all. But it wasn't clear, at least that wasn't the clear driver here. It was really much more of the myeloid and TAM phenotypes. And actually the angiogenic tumors that were low were also similarly associated, not quite as clean of a signal, but certainly it looks like if you're a low angiogenic tumor, you're much more likely to recur. So this suggested that these microenvironmental features also might be useful for adjuvant strategies. And indeed, a lot of the work now that's going forward in some of the newer adjuvant trials are factoring in things like these transcriptomic signatures into risk-adapted models. And I think, the future, of course, would incorporate some of these into selecting not only who's going to recur or not, but maybe whether you give them a combination strategy or a single agent drug. But how can we test this? So ultimately, I show a lot of nice kind of genomic correlative work and maybe some evolution of how we think about kidney cancer from a microenvironmental and genomic standpoint, but really how did we test this? And so this was the challenge that I faced. I was kind of writing all these papers and thinking about this a lot and getting advice from mentors, and everyone kept saying, well, you gotta find a model that works. And so, we didn't have a lot of good models at the time. And I'm going to talk about one. We've developed a second, which I think is maybe even better, but I'm going to talk about the first one today. So there are syngeneic models in kidney cancer. The one that was used really until this time was really the Renka model, which is, for those of you familiar, it spontaneously arose in a BALB/C mouse, which is an immunocompetent mouse. And it was called the renal cortical adenocarcinoma. Back then, we really had very limited understanding. But it has been profiled now, and we know that it's not a VHL-driven tumor. So VHL, as I showed you earlier, is the fundamental event in clear cell. You have to have a VHL mutation to be a clear cell, really. And so it was used forever. GEM models, of course, we know the genetics, so why can't we just use GEMs? And there are many, many GEM models. You can probably find 6 or 7 out there. They're often from mixed backgrounds. They have a very long tumor initiation time. Very lower tumor penetrance compared to other GEMs, and they're very hard to transplant. And often they develop cystic renal failure because when you knock out, even in a KSP specific or CRE specific, kidney specific fashion, you'll often develop cystic renal failure, which is what you see in people with hereditary kidney cancers. Many of them, especially with VHL, will develop many cysts in their kidney in addition to tumors. So it's hard to use those models. So we teamed up with Scott Lowe together, and Scott Lowe is, for those of you not familiar with him, is very, very accomplished scientist at Memorial and does a lot of mouse engineering in beautiful ways. And we used an electroporation strategy at the time, which would allow us to deliver guides. We focused on an actually interesting combination of genes, which are not super common in kidney cancer, but they define, and I'll show you in a minute, some of the myeloid phenotypes, which is one of the reasons why we focus on it. There also happened to be a very nice gem from Ian Fru's group in Germany at the time that utilized VHL-RB and p53 and showed very nicely a gem that we had been using in the lab for many years, and I'll show you some of that work in a minute. But we had utilized this strategy because Scott had been utilizing p53 in a lot of different tumor models, and he had very good guides for it and very good strategies, but also because of the genetic combination and also because of the myeloid phenotype. So this was sort of our strategy at the time. And this was not trivial because we had to have VHL as a backbone, and that makes cells very tricky to add additional guides. For some reason, when you knock out VHL in vitro, the cells don't tolerate it very well. They senesce. And so this was about a year and a half of work that Lynn did. And eventually, though, we were able to develop a tumor that we could transplant, and we were able to show transcriptomically that this matched into that myeloid high group that Bob Motzer and others from Genentech had shown to be critical for that cluster, the very aggressive one that seems to be resistant to VEGF. And I can show you here, this was that cluster here. It's p53 enriched, 30%. Again, p53 is not commonly seen in localized kidney cancer, but if you look at metastatic cohorts, it's about between 10 and 30% of those will have them. And so this is really the stromal proliferative cluster. When you do flow cytology analysis on it, they are very macrophage-enriched tumors, and they have a p53 program when you look at transcriptomics, very similar to that group. And as I mentioned, there was this adenosine signature that we saw, which overlapped with the myeloid signature. This actually came from this paper that was published from UCSF, from Fong et al. In combination with Corvus, which is a biotech company, and they had developed an adenosine 2 receptor blockade therapy for patients with metastatic kidney cancer that were treatment refractory, and they had developed a signature which was very much overlapping with this myeloid signature. So this gave us a thought of, okay, well, we showed that this myeloid program is so important for metastasis development. Maybe if we targeted this adenosine pathway, this could abrogate metastasis. And so what Lynn did in her model when she developed it was started testing CP1444 as this adenosine inhibitor, and we were able to show that you get this dramatic abrogation of metastasis. It doesn't fully control it, but the number of Mets and the development of Mets is abrogated quite dramatically using a myeloid depletion strategy, or a specific myeloid depletion strategy. It doesn't deplete all TAMs, but it does shift the phenotype of some of the TAMs quite dramatically, suggesting perhaps that this could be a strategy in utilizing a mouse model. In the background of all this, we've also, really been thinking about how to utilize the microenvironment to study resistance. And so again, we had to go back to a mouse model, and I showed the syngeneic model, which we developed, but we're in the background of all this for many years. We had been utilizing this gem, and we utilized this gem from Ian Fru et al. This was a KSP, CRE-ERT2 KSP1 Flox mouse that had lost VHL, p53, and RB. They developed pretty nice clear cell tumors. And we utilize this model to actually study some of these questions. We want to understand what's happening with both PD-1 and VEGF therapies alone and in combination. Again, I showed you the relevance of this from a predicting response standpoint. And so we first looked at these tumors genomically. This is unpublished data, but hopefully will be submitted in the next few months. So we started looking at these KVPR tumors that had, from this Ian Fru model, again, validating the fact that they are really representative of this myeloid high tumor that I had shown you earlier from Bob's work. This is, again, showing RNA sequencing from many tumors that we had from these mice, and that they overlap very nicely with the myeloid high p53-driven tumors in human. And we started treating these mice and really focusing on a combination strategy, which I think is near to dear to Harriet from her work in melanoma. But we utilized lenvatinib and PD-1 in combination for a few reasons. One, we were very interested in lenvatinib and PD-1 combo. That has the highest overall response rate. If you look at the clinical trials, it's about 75% of patients will have a first-line response, which is really, really incredible. And also, we know that Lenva has potentially a lot of microenvironmental targets beyond just VEGF. So we were very interested in this question. And we utilized a sort of a mouse clinical trial from this work. We also included, though, a CSF1 inhibitor, BLZ, 945 to see if we just broadly depleting TAMs would be helpful. And I should note that CSF1 and R inhibitors have been a dud in the clinic, primarily because they tend to deplete lots of TAMs, and TAMs can be good and they can be bad. So we didn't really know what to expect here. And I'll just show some of that data. And we did single cell on pretty much all of these mice that we developed tumors from in different categories. And this model actually is quite sensitive to lenvatinib. And actually the combination is pretty dramatically responsive here, but they don't respond at all to PD-1. And actually CSF1 inhibitors don't really do anything at all. So we were then also able to take early responders and resistant and actually start comparing them as well. So we can look at the impacts on the microenvironment from these different treatment strategies alone, in combination, and in resistance and in sensitivity, which is really, I think, something you want to do. And we could take single cell data from this same strategy that we applied before and start looking at the differences between responders and non-responders, between ones that are in combination or alone, and get a sense of what's really driving this. You know, one of the interesting things about this single cell data set was that we actually had a lot of neutrophil populations, and we don't really know their role. I mean, there's been some really nice work from Taha Margoob and Jed Walchuk recently on neutrophils' roles in immunotherapy strategies. So it's an emerging thing, but I'm not going to talk about that too much today, but it was a striking finding here. And we could further subtype the macrophage clusters within here and understand what's happening. It's a very TAM-dominated tumor type, as I mentioned, with the P53 and RB. And we can actually further sub-stratify them and to understand what's actually happening in the context of both therapeutic sensitivity and resistance. And you can start to see that certain TAMs are associated with response and certain TAMs are associated with resistance. So even though if you just deplete all TAMs, you would actually lose that effect. But actually, if you understood which TAMs, which tumor-associated macrophages are actually associated with response, these angio-high TAMs that are producing angiogenic genes, which maybe have been reflected by some of those bulk RNA sequencing data earlier, they're actually associated with response, whereas other TAMs, maybe those myeloid-high, angio-high tumors that don't respond, are actually associated with resistance. So now we can start getting further into the phenotypes of these TAMs and the context of treatment strategies. And for the sake of time, I won't talk about the neutrophils, but it is another story. And we can actually use neutrophils to associate, again, further responses. So perhaps there's a major role for them, and I don't have time to talk about it. But then of course, you have to go back to the human, right? Because I showed you something in mouse, but are there analogous populations in human post-treatment, right? Because if you can cure lots of mice, but you don't know if those populations are relevant to human biology, and that's a major challenge for immunogenomic studies or immunotherapy-related studies, because of course, the mouse and the human microenvironments can be quite different. So that requires post-treatment tissue. So how do you do that? So that's, some of the beauty about going back and forth in my group. So this is work that was led by Stephen Reese, who's graduating from our program this year. He spent a year with me in the lab, and a whole host, including a lot of very talented pathologists and research pathologists, and of course, Christina Leslie from the computational biology. And we took all these patients that we've been operating on over the years now, and we started to define them into categories, right, of early response, of complete response, partial response, and no response. These are patients that Pat operates on all the time, and I operate on quite a bit. And these are patients that are post-immunotherapy now, which is a new frontier. A lot of our surgery now is in the post-IO space. That gives you a very, very unique opportunity to actually study what's happening both within and across tumors. And so this allows us to assemble cohorts of patients that have been exposed to IO therapy alone, to IO TKI therapy, and we have some with just TKI alone, but that's really not done anymore. So essentially, we can look at responses, both defined clinically, radiographically, but also pathologically, and understand what are the populations in human, and are they analogous to the mouse, of course, which is something that I really want we really want to focus on. And so you can start defining this different ways. There's not an official canon here on how to do this. It's sort of been adopted a lot from the melanoma literature about how to define true pathologic response. A lot of us have looked at, complete response where there's no residual viable tumor, RVT, residual viable tumor, or near complete response. And those patients actually, if you take their kidneys out, we showed and we'll show in our paper that, they have really, really durable responses, many, many years without even off therapy. So it's a good biomarker for how they're going to do down the road. And then you can have these partial responses where they have this in between, and you have these non-responses where there's really no treatment response that you can see at all within the tumor. And you could do single cell sequencing on these cohorts and start to get, obviously in the human, you still see a lot more T cells, as I showed you earlier from the work that we did and what David has done. But you can see these TAM populations here, and then you can overlay work that Andrew Cornish, who's an MD medical oncology fellow working with Ming Li, has done a lot of this work now, and we can start seeing what are the differences between the IO only and the IO TKI and the untreated populations in terms of the single cell, again, post-treatment populations, and we can start focusing on some of these same populations that we saw, and then we can actually overlay the mouse TAM signatures onto these populations to see, are there analogous populations and are they associated with resistance and response, both within the tumors and across the different regions. And that's sort of where we're focusing now. And so we can further substratify the TAMs, just like I showed you in the mouse, and to see, are the TKIs really depleting some of these? This is just looking at them broadly without looking at resistance, but you can start seeing that the different populations are being affected in different ways by the different treatments, in maybe similar ways, but I'm sure different ways as well as the mouse. But hopefully that will help us hone in on what are the most relevant targets to try in the mouse. And you could see this, if you focus on one of the M0 signatures from the mouse that I showed you, that gem mouse, you could see that there's clearly differences in terms of the TKI-IO combination patients. And the upper tail and the lower tail on this plot are actually associating with resistance and response. So you get a sense that maybe these populations are relevant across species. So that, I think, is sort of where we're headed. So overall, my conclusions really are that RNA signatures and immune response are really the useful ones clinically. I showed you sort of the genetic recap of kidney cancers and how it might relate to some of these microenvironmental features, but that's really what we have from a predictive and prognostic standpoint. And maybe it'll help us select adjuvant treatment strategies down the road for patients. Certainly pick the high-risk patients a little bit better, perhaps. The phenotype seems to be enriched in the metastatic setting, and particularly post-IO, and maybe this cross analysis will allow us to prioritize targets to test preclinically, and then hopefully bring them out to the clinic now that more and more companies are interested in targeting TAM populations with different inhibitors. And I want to obviously thank my funding and, of course, members of my lab, Ming Li's lab, the urology department, Christina Leslie from computational biology, and all the medical oncologists I work with, and particularly Dr. Motzer, who's been a wonderful mentor to me for many, many years. Thank you so much. And I'll have you answer any questions.
Thanks, Ari. That was a real tour de force. I have a question about the complexity of your clustering. So I noted that you had 21 clusters of myeloid cells in one of your figures. I believe it was one of the mouse figures.
Yeah, well, so that's sort of the art and the dark art of single cell sequencing. You can cluster any way you want, and you can set your parameters quite differently. So yeah, I mean, this is all myeloid populations. You're absolutely right. So one of the things we do then, of course, is then go back with my immunology colleagues and actually start to think about what are really representing unique populations versus just slicing and dicing single cell data in more and more complex ways. And we try to validate them by flow and to look at really the dominant populations there. So it's just this is just sort of an early iteration of what would be a real clustering.
No, I get it. And it is really complicated before treatment, on treatment. But my other question is, spatially, are some of the clusters uniquely positioned in a certain area of the large tumors? That's in humans, I guess, is where I'm interested.
Yeah. So in that same cohort that I showed you that we're working with Hartland Jackson, who's in Toronto, who's a spatial mass cytometry kind of person. We developed from the single cell data a series of populations, really relying on mostly the human. So we took a conglomerate of the different single cell studies that David has done and others have done, and kind of come up with like a meta-analysis of what are the key markers of the different TAM populations to reduce it down to maybe 5 or 6 that might, you tag a TAM by, CD68 or something else, and then you can add a few additional markers and then look at the spatial orientation in these contexts. So we're taking all these regions both within tumors and across tumors and reducing it down to probably a core. And I think ultimately from a biomarker standpoint, you want to kind of just be able to choose a particular TAM or a particular T cell that would be relevant and just reduce it to a couple quick stains that a pathologist could hopefully do, as opposed to have to do fancy and very expensive sequencing. So absolutely thinking about the same questions that you bring up.
Thank you. I guess I haven't. Not, I got an unrelated one.
Sure.
Completely unrelated. The mouse cell line. So thank you for sending us and sharing that
Of
mouse
course.
cell line with us. Are you planning on making additional ones with different genetic
Yeah,
perturbations?
yeah. So
That's real community service here. So
yeah, so we are doing we have a VHL BAP1 CDKN2B, which is a more common combination, and that one is a sarcomatoid tumor, perfectly responds well to CTLA-4 very nicely. So that one will be, hopefully that paper's, we're finishing up this that work, but I think that will be something that people find more exciting just because of the common genetics. In fact, when I presented this one initially, Bill Kalin very astutely said, that's not a very common genetic strategy. I'm like, yeah, but it's a common one for the bad tumors that don't respond well. So this one is a much more common genetic subtype, and it's a challenge of doing any of these engineering. We tried, of course, all the more common mutations, as has Bill and others. Chris bring out PBRM1. The tumors just don't grow well. They're very hard. So the nice clear cells are hard to engineer. The bad ones that don't look super clear cell but they retain the CA9 and HIF1 at least, don't look. Those are the ones that grow. It's a challenge of any syngeneic system. So that's why you can rely on GEMs, but GEMs are just hard to treat. So limitation of the field. Sure.
Thank you. So really great work in terms of single cell transcriptomics and immune profiling and CyTOF and such. But ultimately, the biomarker should be translated into clinic and should be easily performed and replicable and cheap. So how do you envision translating these signatures into the clinic?
That's great. So a couple ways, and I think we're thinking about this a few different ways. So one, of course, is where obviously reducing it to a few markers that might stain for the most relevant populations. And maybe it's a combination of TAMs, maybe some neutrophils and some CD8 populations that might ultimately come up with a very straightforward IHC panel. The other thing, of course, is that what a lot of people are thinking about is, digital pathology and sort of AI. So if you can define groups of tumors transcriptionally, and then you put it into some model where you have the scan slide scanned in and put through a machine learning platform, you could maybe even digitally say this tumor has this feature, even though the pathologist has no idea what they're seeing, but the model does. And so for that, and I know a lot of people are working on this, but for that same Novartis study where we showed that the myeloid phenotype is associated with recurrence. We're working with a group at Dartmouth that has a machine learning model. We have all the transcriptomic. We have the slides sent from Novartis, which is like 12 terabytes of data. They high resolution scanned all the slides from that trial, and we've given them the microenvironmental subgroups and strategies, and they're trying to figure out if they can do that. And they have already done it on the TCGA, so you can kind of replicate it, because that might be a way to say, okay, now you put it through an AI system, and they say this tumor is this thing, and this patient's going to recur much more, or this patient might respond better. So that's a strategy that I think a lot of us are thinking about. Great question. Okay.
For the community service and making these mouse models, and thank you for sharing with us as well. I'm curious, from the immuno-oncology metabolism world, we talk a lot about the obesity paradox in melanoma and lung cancer. Do patients with obesity respond better to immunotherapy? And so I'm wondering if knowing that RCC actually is associated with obesity, I'm wondering if you can speak to your mouse models, if you've observed any potential difference in the response to immunotherapy in these models in mice with obesity, and if not, maybe we could collaborate on that.
Yeah, so it's a great question, and something that's very near and dear. So I do have funding through the DoD to look at obesity in kidney cancer and these models, and we've been utilizing the transplantation models. We did the GEM model first. We did fat feed them. They're hard to feed because of the mixed background, so they gain weight not as nicely as if they were a clean genotype. But we do observe earlier onset tumors in those mice. But then syngenetically, when we implant them, and that's not trivial, by the way, if you're going to do an orthotopic transplantation model in a fat mouse, because just like humans, they develop a lot of perinephric fat. So if you try to open up the mouse and inject it, it's like a shit show, pardon my French. But essentially, it's very challenging. So we've been doing, but we do see that the tumors grow faster in obese, which sort of makes sense because we know that obesity is associated with better development of kidney cancers. But it does suggest, in human at least, they seem to be less aggressive. So we're now trying to understand immunologically what's going on. But I think we're talking soon. So I'm happy to talk more about that, Rachel. But I think it would be it's a really cool area. And we're focused on TREM2 macrophages, which has been shown to be associated with the lipid-associated macrophages, and it's associated with more aggressive tumors. So we can talk more about that, but definitely something that we're thinking a lot about.